Hello students, today we will learn about enzyme technology. Enzymes are the biocatalysts synthesized by living cells. They are complex protein molecules that bring about chemical reactions concerned with life. In it is fortunate that enzymes continue to function when they are separated from the cells, that is in vitro. Basically, enzymes are non toxic and biodegradable. They can be produced in large amounts by microorganisms for industrial applications. Enzyme technology broadly involves production, isolation, purification, and use of enzymes for the ultimate benefit of humankind. In addition, recombinant DNA technology and protein engineering involved in the production of more efficient and useful enzymes are also a part of enzyme technology. The commercial production and use of enzymes is a major part of biotechnology industry. The specialties like microbiology, chemistry and process engineering besides biochemistry have largely contributed for the growth of enzyme technology. Now let's know about the applications of enzymes. Enzymes have a wide range of applications. These include their use of food production, food processing, preservation, washing powders, textile manufacturing, leather industry, paper industry, medical applications, improvement of environmental and the scientific research. As per the recent estimates, a great majority of industrially produced enzymes are useful in processes related to foods, that is maybe 45%, detergents that may be 35%, textiles that may be 10%, and leather that may be 3%. Now let's know about the commercial production of enzymes. Microbial enzymes have been utilized for many centuries without knowing them fully. The first enzyme produced industrially was Decadiester diastase in it's a fungal amylase in 1896 in United States. It was used as a pharmaceutical agent to cure digestive disorders. In Europe, there existed a centuries old practice of softening the hides by using feces of dogs and pigeons before tanning. A German scientist demonstrated in 1905 that extracts from animal organs, that pancreas from pig and cow, could be used as a source of enzymes proteases for leather softening. The utilization of enzymes, chiefly proteases, for laundry purposes started in 1915. However, it was not continued due to allergic reactions of impurities in enzymes. Now, special techniques are available for manufacture and use of enzymes in washing powders without allergic reactions. Commercial enzymes can be produced from a wide range of biological sources. At present, a great majority of 80% of them are from microbial sources. The different organisms and their relative contribution for the production of commercial enzymes are fungi, that is 60%, bacteria, that is 24%, yeast 4%, streptomyces 2%, higher animals 6%, and higher plants 4%. A real breakage for large scale industrial production of enzymes from microorganisms occurred after 1950s. Enzymes from animal and plant sources. Let's know about enzymes from animal and plant sources. In the early days, animal and plant sources largely contributed to enzymes. Even now, for certain enzymes, they are the major sources. Animal organs and tissues are very good sources for enzymes such as lipases, esterases, and proteases. The enzyme lysozyme is mostly obtained from hen eggs. Some plants are excellent sources for certain enzymes, papain, that is from extracted from papaya, and bromelain, it's extracted from pineapple. Now let's know about the limitations. There are several drawbacks associated with the manufacture of enzymes from animals and plant sources. The quantities are limited and there is a wide variation in their distribution. The most important limitations are the difficulties in isolating, purifying, and enzymes and the cost factor. As regards extraction of industrial enzymes from bovine sources, there is a heavy risk of contamination with bovine spongiform encephalopathy. For this reason, microbial production of enzymes is preferred. Enzymes from mammalian cell cultures. Let's know about enzymes from mammalian cell cultures. There exists a possibility of producing commercial enzymes directly by mammalian cell cultures, but the main constraint will the cost factor, which will be extremely high. However, certain therapeutic enzymes such as tissue plasminogen activator are produced by cell cultures. Now let's know about the enzymes from microbial sources. Microorganisms are the most significant and convenient sources of commercial enzymes. They can be made to produce abundant quantities of enzymes under suitable growth conditions. Microorganisms can be cultivated by using inexpensive media, and production can take place in a short period. In addition, it is easy to manipulate microorganisms in genetic engineering to increase the production of desired enzymes. Recovery, isolation, and purification processes are easy with microbial enzymes than that with animal and plant sources. In fact, most enzymes of industrial applications have been successfully produced by microorganisms. Various fungi, bacteria, and yeast are employed for this purpose. Now let's know about Aspergillus niger, a unique organism for production of bulk enzymes. Among the microorganisms, Niger or Aspergillus niger, it's a fungus, occupies a special position for the manufacture of a large number of enzymes in good quantities. There are well over 40 commercial enzymes that are conveniently produced by A. niger. These include alpha amylase, cellulase, protease, lipase, pectinase, phytase, catalase, and insulinase. Now let's know about the technology of enzyme production general considerations. In general, techniques employed for microbial production of enzymes are comparable to the methods used for manufacture of other industrial products. Now let's know about some features. It's a selection. First point is selection of organisms. Second is formulation of medium. Third is production process. Fourth is recovery and purification of enzymes. Now let's know about selection of an organism. The most important criteria for selecting the microorganism are that the organism should produce the maximum quantities of desired enzyme in a short time, while the amounts of other metabolite produced are minimal. Once the organism is selected, stain improvement for optimizing the enzyme production can be done by appropriate methods. From the organism chosen, inoculum can be prepared in a liquid medium. Now let's know about formulation of medium. The culture medium chosen should contain all the nutrients to support adequate growth of microorganisms that will ultimately result in good quantities of enzyme production. The ingredients of medium should be readily available at low cost and are nutritionally safe. Some of the commonly used of substrates for the medium are starch hydrolysate, molasses, corn starch liquor, yeast extract, whey, and soybean meal. Some cereals and pulses have also been used. The pH of the medium should be kept optimal for good microbial growth and enzyme production. Let's know about the production process. Industrial production of enzyme is mostly carried out by submerged liquid conditions and to a lesser extent by solid substrate fermentation. In submerged culture technique, the yields are more and chances of infection are less. Hence, there is a preferred method. However, solid substrate fermentation is historically important and still in use for the production of fungal enzymes. Example: amylases, cellulases, proteases, and pectinases. The medium can be sterilized by employing batch or continuous sterilization techniques. The fermentation is started by inoculating the medium. The growth condition, that is pH, temperature, oxygen supply, nutrient addition, are maintained at optimal levels. The froth formation can be minimized by adding antifoam agents. The production of enzymes is mostly carried out by batch fermentation and to a lesser extent by continuous process. The bioreactor system must be maintained sterile throughout the fermentation process. The duration of fermentation is variable around 2 to 7 days. In most production processes besides desired enzymes, several other metabolites are also produced. The enzyme have to be recovered and purified. Now let's know about recovery and purification of enzymes. The desired enzyme produced may be excreted into the culture medium or may be present within the cells. That means intracellular enzymes. Depending on the requirement, the commercial enzyme may be crude or highly purified. Further, it may be in the solid or liquid form. The steps involved in downstream processing, that is recovery and purification steps employed, will depend on the nature of the enzyme and the degree of purity desired. In general, recovery of an extracellular enzyme which is present in the broth is relatively simpler compared to an intracellular enzyme. For the release of intracellular enzymes, special techniques are needed for cell disruption. The physical, chemical, and enzymatic methods adopted to break the cells and release their contents are now we will discuss. We discussed already. The reader must be invariable. Refer them invariable. Microbial cells can be broken. Microbial cells can be broken down by physical means. That means sonication, high pressure glass beads. The cell wall of bacteria can be lysed by enzyme lysozyme. For yeast, the enzyme beta-glucanase is used. However, enzymatic methods are expensive. The recovery and purification steps will be the same for both intracellular and extracellular enzymes. Once the cells are disrupted and intracellular enzymes are released, the most important consideration is to minimize the loss of desired enzyme activity. Now let's know about removal of cell debris. Filtration or centrifugation can be used to remove cell debris. Now let's know about removal of nucleic acids. Nucleic acids interfere with the recovery and purification of enzymes. They can be precipitated and removed by adding polycations such as polyamines, streptomycin, and polyethylene imine. Now let's know about enzyme precipitation. Enzymes can be precipitated by using salts, aminosulfate, ammonium sulfate, organic solvents, that is like isopropanol, ethanol, acetone. Like this, precipitation is advantageous since the precipitated enzyme can be dissolved in a minimal volume to concentrate the enzyme. Now let's know about liquid-liquid partition. Further concentration of desired enzymes can be achieved by liquid-liquid extraction using polyethylene glycol or polyamines. Now let's know about separation by chromatography. There are several chromatographic techniques for separation and purification of enzymes. These include ion exchange, size exclusion, affinity, hydrophobic interaction, and dye-ligand chromatography. Among these, ion exchange chromatography is the most commonly used for enzyme purification. Now let's know about drying and packing. The concentrated form of the enzyme can be obtained by drying. This can be done by film evaporators or freeze dryers. The dried enzyme can be packed and marketed. For certain enzymes, stability can be achieved by keeping them in ammonium sulfate suspensions. All the enzymes used in foods or medical treatments must be of high grade purity and must meet the required specifications by regulatory big bodies. These enzymes should be totally free from toxic materials, harmful microorganisms, and should not cause allergic reactions. Now let's know about the regulation of microbial enzyme production, that is general considerations. A maximal production of microbial enzymes can be achieved by optimizing the fermentation conditions, that may be nutrition, nutrient, pH, oxygen, temperature, etc. For this purpose, a clear understanding of the genetic regulation of enzyme synthesis is required. Some of the general aspects of microbial enzyme regulation now we will discuss. First is induction. Several enzymes are inducible, that is, they are synthesized only in the presence of inducers. The inducer may be the substrate or product or intermediate. The inducer compounds are expensive and their handling, like you have to sterilize, addition of at specific time, also is quite difficult. In recent years, attempts are being made to develop mutants of microorganisms in which inducer dependence is eliminated. Now, let's know about the feedback. Feedback regulation by the end product significantly influences the enzyme synthesis. This occurs when the end product accumulates in large quantities. Large scale production of feedback regulated enzymes is rather difficult. However, mutants that lack feedback repression have been developed to overcome this problem. Now let's know about the nutrient repression. The native metabolism of microorganism is so divided that there occurs no production of unnecessary enzymes. In other words, the microorganisms do not synthesize enzymes that are not required by them. Since this is a wasteful exercise, the inhibition of unwanted enzyme production is done by nutrient repression. The nutrients may be carbon, nitrogen, phosphate, or sulfate supplies in the growth medium. For large scale production of enzymes, nutrient repression must be overcome. Now let's know about glucose repression. Glucose repression is a classical example of nutrient repression, more appropriately catabolite repression. That this is in the presence of glucose and the enzyme needed for the metabolism of rest of the compounds are not synthesized. Glucose repression can be overcome by feeding of carbohydrate to the fermentation medium in such a way that the concentration of glucose is almost zero at any given time. In recent years, attempts are being made to select mutants that are resistant to catabolite repression by glucose. In certain microorganisms, other carbon sources such as pyruvate, lactate, citrate, and succinate also act as catabolite repressors. Now, let's know about nitrogen source repression. Nitrogen source is also observed in microorganisms. This may be due to ammonium ions and amino acids. Most commonly inexpensive ammonium salts are used as nitrogen sources. The repression by ammonium salts can be overcome by developing mutants resistant to these nitrogen sources. Now let's know about genetic engineering for microbial enzyme production. Enzymes are the functional products of genes. Therefore, theoretically, enzymes are good candidates for improved production through genetic engineering. During the past 15 years, the advances in the recombinant DNA technology have recently helped for increasing the microbial production of commercial enzymes. It is now possible to transfer the desired enzyme genes from one organism to the other. Once an enzyme with a potential use in industry is identified, the relevant gene can be cloned and inserted into a suitable production host. Let's know about cloning strategies. This involves the development of cDNA library for the mRNA and creation of oligonucleotide probes for the desired enzyme. On hybridization with oligonucleotide probes, the specific cDNA clones can be identified. The next step is the transformation of industrially important host organism, example Aspergillus oryza, for the production of the desired enzyme. By this approach, it is possible to manufacture high quality industrial enzymes. A couple of enzymes produced by employing cloning strategies we will now discuss. First is the enzyme lipolase. The enzyme lipolase found in the fungus Humicola lang- languinosa Humicola languinosa is very effective to remove fat stains in fabrics. However, industrial production of lipolase by this organism is not possible due to a very low level of synthesis. The gene responsible for lipolase was isolated, cloned, and inserted into Aspergillus oryza. Thus, large-scale production of this enzyme was successfully achieved. Lipolase is very stable and resistant to degradation by proteases that are commonly used in detergents. All these properties make lipolase a strong candidate for in its use fabric washing. Rennet chymosin. Rennet is an enzyme widely used in making cheese. It is mainly obtained from the stomach of young calves. Consequently, there is a shortage in its supply. The genes for the synthesis of chymosin has been cloned for its large-scale production. Protein. Let's know about protein engineering for modification of industrial enzymes. It is now possible to alter the structure of a protein or enzyme by protein engineering and site-directed mutagenesis. The changes in the enzymes are carried out with the objective of increased enzymes stability and its catalytic function. Resistant to oxidation change substrate preference, and increased tolerance to alkali and organic solvents. By site-directed mutagenesis, selected amino acids are at specific positions can be changed to produce an enzyme with desired properties. For instance, protein engineering has been used to structurally modify phospholipase A2 that can resist a high concentration of acid. The modified enzyme is more efficiently used as a food emulsifier. Genetic engineering has tremendous impact on the industrial production of enzymes with desired properties in a cost-effective manner. Now let's know about immobilization of enzymes and cells. Traditionally, enzymes in free solutions, that is insoluble or free form, react with substrates to result in products. Such use of enzymes is wasteful, particularly for industrial purposes, since enzymes are not stable and they cannot be recovered for reuse. Immobilization of enzymes refer to the technique of confining or anchoring the enzymes in or on an inert support for their stability and functional reuse. By employing this technique, enzymes are made more efficient and cost effective for their industrial use. Some workers regard immobilization as a goose with a golden egg in enzyme technology. Immobilized enzymes retain their structural conformation necessary for catalysis. There are several advantages of immobilized enzymes. Example one first is stable and more efficient in function. Second is can be reused again and again. Third is ideal for multi-enzyme reaction systems. Fourth is control of enzyme function is easy. Fifth is suitable for industrial and medical use. Sixth is minimize effluent disposal problems. There are, however, certain disadvantages also associated with immobilization. The possibility of loss of biological activity is an enzyme Immobilization or while it is in use. Immobilization is an expensive affair, often required sophisticated equipments. Immobilized enzymes are generally preferred over immobilized cells due to specificity to yield the products in pure form. However, there are several advantages of using immobilized multi-enzyme systems such as organelles and whole cells over immobilized enzymes. The immobilized cells possess the natural environment with cofactor availability and are particularly suitable for multiple enzymatic reactions. Now let's know about the methods of immobilization. The commonly employed techniques for immobilization of enzymes are adsorption, entrapment, covalent binding and cross-linking. Now let's know about adsorption. Adsorption involves the physical binding of enzymes on the surface of an inert support. The support materials may be inorganic, example alumina, silica gel, calcium phosphate gel, or organic, that is starch, carboxymethyl cellulose, DEAE cellulose, DEAE sephadex. Adsorption of enzyme molecules on the inert support involves weak forces such as van der Waals forces and hydrogen bonds. Therefore, the adsorbed enzymes can be easily removed by minor changes in pH, ionic strength, or temperature. This is a disadvantage for industrial use of enzymes. Now let's know about entrapment. Enzymes can be immobilized by physical entrapment inside a polymer or a gel matrix. The size of the matrix pores is such that the enzyme is retained while the substrate and product molecules pass through. In this technique, commonly referred to as lattice entrapment, the enzyme is not subjected to strong binding forces and structural dissertations. Some deactivation may, however, occur during immobilization process due to changes in pH and temperature and or addition of solvents. The matrices used for entrapping the enzymes include polyacrylamide gel, collagen, gelatin, starch, cellulose, silicon, and rubber. Enzymes can be entrapped by several ways. First is enzyme inclusion in gels. This is an entrapment of enzymes inside the gels. Second is enzyme inclusion in fibers. The enzymes are trapped in a fiber format of the matrix. Third is enzyme inclusion in microcapsules. In this case, the enzymes are trapped inside a microcapsule matrix. The hydrophobic and hydrophilic forms of matrix polymerize to form a macrocapsule containing enzyme molecules inside. The major limitations for entrapment of enzymes is their leakage from the matrix. Most workers prefer to use the technique of entrapment for immobilization of whole cells. Entrapment cells are in use for industrial production of amino acids, that is L isoleucine, L aspartic acid, L malic acid and hydroquinone. Now let's know about microencapsulation. Encapsulation, microencapsulation. Microencapsulation is a type of entrapment. It refers to the process of spherical particle formation wherein a liquid or suspension is enclosed in a semi-permeable membrane. The membrane may be polymeric, lipodial, lipoidal, lipoprotein based or non-ionic in nature. There are three distinct ways of microencapsulation. First is building of specific membrane reactors. Second is formulation of emulsions. Third is stabilization of emulsions to form microcapsules. Microencapsulation is recently being used for immobilization of enzymes and mammalian cells. For instance, pancreatic cells growth in cultures can be immobilized by microencapsulation. Hybridoma cells have also been immobilized successfully by this technique. Now let's know about covalent binding. Immobilization of the enzymes can be achieved by creation of covalent bonds between the chemical groups of enzymes and the chemical groups of the support. This technique is widely used. However, covalent binding is often associated with loss of some enzyme activity. The inert support usually requires pre-treatment to form pre-activated support before it binds to enzymes. Now let's know about cyanogen bromide activation. The inert support materials, that is cellulose, separose, sephadex containing glycol groups are activated by CNBR, which then bind to enzymes and immobilize them. Now let's know about diazotation. Some of the support materials: aminobenzyl cellulose, amino derivatives of polystyrene, amino salinized porous glass are subjected to diazotation on treatment with NaNO2 and HCl. They in turn bind covalently to tyrosyl or histidyl groups of enzymes. Now let's know about third is peptidyl bond formation. Enzyme immobilization can also be achieved by the formation of peptide bonds between the amino groups of support and the carboxyl groups of enzymes. The support material is first chemically treated to form active functional groups. Now let's know about activation by bi or polyfunctional reagents. Some of the reagents such as glutaraldehyde can be used to create bonds between amino groups of enzymes and amino groups of support. Example: amino ethyl cellulose, albumin, amino alkylated porous glass. Now let's know about cross-linking. The absence of a solid support is a characteristic feature of immobilization of enzymes by cross-linking. The enzyme molecules are immobilized by creating cross-linking between them. Therefore, through the involvement of polyfunctional reagents. These reagents in fact react with the enzyme molecules and create bridges which form the backbone to hold enzyme molecules. There are several reagents in use for cross-linking. These include glutaraldehyde, diisobenzidine, hexamethylene, diisocyanate and toluene diisothiocyanate. Glutaraldehyde is the most extensively used cross-linking reagent. It reacts with lysyl residues of the enzymes and forms a base. The cross-links formed between the enzyme and the glutaraldehyde are irreversible and can withstand extreme pH and temperature. Glutaraldehyde cross-linking has been successfully used to immobilize several industrial enzymes, example glucose isomerase, penicillin amidase. The technique of cross-linking is quite simple and cost-effective, but the disadvantage is that it involves the risk of denaturation of the enzyme by polyfunctional reagent. Now let's know about choice of immobilization technique. The selection of a particular method for immobilization of enzymes is based on a trial uh, and error approach to choose the ideal one. Among the factors that decide a technique, the enzyme catalytic activity, stability, regenerability, and cost factor are important. Now let's know about immobilization of L-amino acylase. L-amino acid acylase was the first enzyme to be immobilized by a group of Japanese workers, Chibata and Tosa, in 1969. More than 40 different immobilization methods were attempted by this group. Only 3 of them were found to be useful. They are covalent binding to iodoacetyl cellulose, ionic binding to DEAE sephadex, and entrapment within polyacrylamide gel. Now let's know about stabilization of soluble enzymes. Some of the enzymes cannot be immobilized and have and they have to be used in soluble form. Examples: enzymes used in liquid detergents, some diagnostic reagents, and food additives. Such enzymes can be stabilized by using certain additives or by chemical modifications. The stabilized enzymes have longer half lives, although they cannot be recycled. Some important methods of enzyme stabilization are now we will discuss. Now let's know about solvent stabilization. Certain Certain solvents at low concentration stabilize the enzymes, while at high concentration the enzymes get denatured. Example: acetone 5% and ethanol 5% can stabilize benzyl alcohol dehydrogenase. Now let's know about substrate stabilization. The active site of an enzyme can be stabilized by adding substrate. Example: starch stabilizes alpha amylose, glucose stabilizes glucose isomerase. Now let's know about stabilization of polymers. Enzymes can be stabilized particularly against increased temperature by addition of polymers such as gelatin, albumin, and polyethylene glycol. Now let's know about stabilization by salt. Stabilization of metalloenzymes can be achieved by adding salts such as calcium, iron, manganese, copper, zinc. Proteases can be stabilized by adding calcium. Now let's know about stabilization by chemical modifications. Enzymes can be stabilized by suitable chemical modifications without loss of biological activity. There are several types of chemical modifications. Addition of polyamino acid chains, polytyrosine, polyglycine. Acylation of enzymes by adding groups such as acetyl, pro- propionyl, succinyl. Now let's know about stabilization by rebuilding. Theoretically, the stability of the enzymes is due to hydrophobic interactions in the core of the enzyme. It is therefore proposed that enzymes can be stabilized by enhancing hydrophobic interactions. For this purpose, the enzyme is first unfolded and then rebuilt in one way. Let's know about the ways. What are the ways to know? First is the enzyme can be chemically treated and then refolded. How? By the help of urea and disulfide. Second is the refolding can be done in presence of low molecular weight ligands. Third is for certain enzymes, refolding at higher temperature stabilizes them. It is around 50 degrees centigrade. Stabilization by site-directed mutagenesis. Site-directed mutagenesis has been successfully used to produce more stable and functionally more efficient enzymes. Example: subtilization E. For, let's know about immobilization of cells. Immobilized individual enzymes can be successfully used for single-step reactions. They are, however, not suitable for multi-enzyme reactions and for the reactions requiring cofactors. The whole cells or cellular organelles can be immobilized to serve as multi-enzyme systems. In addition, immobilized cells rather than enzymes are sometimes preferred even for single reactions due to cost factor in isolating enzymes for the enzymes which depend on the special arrangement of the membrane. Cell mobilization is preferred. Immobilization Immobilized cells have traditionally used for the treatment of sewage. The techniques employed for immobilization of cells are almost the same as that used for immobilization of enzymes, with appropriate modifications. Entrapment and surface attachment techniques are commonly used. Gels and some extend membranes are also employed. Now let's know about immobilized via viable cells. The viability of the cells can be preserved by mild immobilization. Such immobilized cells are particularly useful for fermentation. Sometimes mammalian cell cultures are made to function as immobilized viable cells. Now let's know about immobilized non-viable cells. In many instances, immobilized non-viable cells are preferred over the enzymes or even the viable cells. This is mainly because of the costly isolation and purification processes. The best example is the immobilization of cells containing glucose isomerase for the industrial production of high fructose syrup. Now let's know about the limitations of immobilizing eukaryotic cells. Prokaryotic cells are mainly Prokaryotic cells are mainly used for immobilization. It is also possible to immobilize eukaryotic plant and animal cells. Due to the presence of cellular organelles, the metabolism of eukaryotic cells is slow. Thus, for the industrial production of biochemicals, prokaryotic cells are preferred. However, for the production of complex proteins, example immunoglobulins, and for the proteins that undergo post-translational modifications, eukaryotic cells may be used. Now let's know about effect of immobilization on enzyme properties. Enzyme immobilization is frequently associated with alterations in enzyme properties, particularly kinetic properties of enzymes. Now we will discuss. In there there is a substantial decrease in the enzyme specificity. This may be due to conformational changes that occurs when the enzymes gets immobilized. Second point is the kinetic constants like Km and V max of an immobilized enzyme differ from that of the native enzyme. This is because the conformational change of the enzyme will affect the affinity between enzymes and substrate. Now let's know about immobilized enzyme reactions. The immobilized enzyme cells are utilized in the industrial processes in the form of enzyme reactors. They are broadly of two types. First is batch reactors, second is continuous reactors. Now let's know about batch reactors. In batch reactors, the immobilized enzymes and substrates are placed and the reaction is allowed to take place under constant stirring. As the reaction is completed, the production is separated from the enzyme and usually by denaturation. Soluble enzymes are commonly used in batch reactors. It is rather difficult to separate the soluble enzymes from products, hence there is a limitation of their reuse. However, special techniques have been developed for recovery of soluble enzymes, although this may result in loss of enzyme activity. Now let's know about stirred tank reactors. The simplest form of batch reactor is the stirred tank reactor. It is composed of a reactor fitted with a stirrer that allows good mixing and appropriate temperature and pH control. However, there may occur loss of some enzyme activity. A modification of stirred tank reactor is basket reactor. In this system, the enzyme is retained over the impeller blade. Both stirred tank reactor and basket reactor have a well mixed flow pattern. Plug flow type reactors. Let's know about the plug flow type reactors. These reactors are alternatively to flow pattern type of reactors. The flow rate of fluid controlled by a plug system. The plug flow type reactors may be in the form of packed bed or fluidized bed, and these reactors are particularly useful when there occurs inadequate product formation in flow type reactors. Further, plug flow reactors are also useful for obtaining kinetic data on the reaction systems. Now let's know about continuous reactors. In continuous enzyme reactors, the substrate is added continuously while the product is removed simultaneously. Immobilized enzymes can also be used for continuous operation. Continuous reactors have certain advantages over batch reactors. These include control over the product formation, convenient operation of the system, and easy automation of the entire process. There are mainly of two types of continuous reactors: continuous stirred tank reactor, that is in short CSTR, and plug reactor in short PR. CSTR is ideal for good product formation. Now let's know about membrane reactors. Several membranes have with a variety of chemical composition can be used. The commonly used membrane materials include polysulfone, polyamide, and cellulose acetate. The biocatalysts are normally retained on the membranes of the reactor. The substrate is introduced into reactor while the product passes out. Good mixing in the reactor can be achieved by using stirrer. In a continuous membrane reactor, the biocatalysts are held over membrane layers on to which substrate molecules are passed. In a recycled model membrane reactor, the contains like that is the solution containing enzymes, cofactors, and substrates along with freshly released product are recycled by using a pump. The product passes out which can be recovered. Now let's know about the applications of immobilized enzymes and cells. Immobilized enzymes and cells are very widely used for industrial, analytical, and therapeutic purposes. Besides their involvement in food production and exploring the knowledge of biochemistry, microbiology, and other allied specialties. Now, let's know about manufacture of commercial products. First is production of amino acids. L-amino acids and not D-amino acids are very important for use in food and feed supplements and medical purposes. The chemical methods employed for their production results in a racemic, racemic mixture of D and L amino acids. They can acylated to form D-L-acyl amino acids. The immobilized enzyme amino acylase, frequently immobilized on DEAE sephadex, can selectively hydrolyze D-L-acyl amino acids to produce L-amino acids. D-L-acyl amino acids in presence of amino acylase gives rise to L-amino acids plus D-L-acyl amino acids. The free L amino acids can separated from the unhydrolyzed D-acyl amino acids. The latter can be racemized to D-L-acyl amino acids and recycled through the enzyme reactors containing immobilized amino acylase. Huge quantities of L-methionine, L-phenylalanine, L-tryptophan, and L-valine are produced widely by this approach. Now let's know about the production of high fructose syrup. Fructose is the sweetest among the monosaccharides and has twice the sweetening strength of sucrose. Glucose is about 75% as sweet as sucrose. Therefore, glucose, the most abundant monosaccharide, cannot be a good substitute for sucrose for sweetening. Thus, there is a great demand for fructose, which is very sweet but has the same calorific, calorific value as that is that of glucose or sucrose. Now let's know about high fructose syrup containing approximately equivalent amounts of glucose and fructose. HFS is almost similar to sucrose from national point of view. HSF, HFS is in brief high fructose syrup is a good substitute for sugar in the preparation of soft drinks, processed foods, and baking. High fructose syrup can be produced from glucose by employing an immobilized enzyme glucose isomerase. The starch containing raw materials, that is wheat, potato, corn, are subjected to hydrolyze to produce glucose. Glucose isomerase then isomerizes glucose to fructose. The product formed is HFS containing about 50% fructose. Glucose isomerase. Let's know about glucose isomerase. This is an intracellular enzyme produced by a number of microorganisms, a species of Arthrobacter, Bacillus, and Streptomyces are preferred sources. Being an intracellular enzyme, the isolation of glucose isomerase without loss of biological activity requires special and costly techniques. Many a times, whole cells of or partly broken cells are immobilized and used. Now let's know about immobilized enzymes and cells analytical applications in biochemical analysis. Immobilized enzymes can be used for the development of precise and specific analytical techniques for the estimation of several biochemical compounds. The principle of analytical assay primarily involves the action of the immobilized enzymes on the substrate. A decrease in the substrate concentration or an increase in the product level or an alteration in the cofactor concentration can be used for the assay. Now, let's know about thermistors. Thermistors are heat measuring devices which can record the heat generated in, in an enzyme catalyzed reactions. Electrode devices are used for measuring potential differences in the reaction system. An enzyme thermistor and an enzyme electrode along with a specific urea electrode. Now we will discuss. Let's know about the affinity chromatography and purification. Immobilized enzymes can be used in affinity chromatography. Based on the property of affinity, it is possible to purify several compounds, example antigen, antibodies, and cofactors. Now let's know about the biosensors. What do you mean by biosensors? A biosensor is an analytical device containing an immobilized biological material, example enzyme, antibody, nucleic acids, hormones, organelles, or whole cells, which can specifically interact with an analyte and produce physical, chemical, or electrical signals that can be measured. An analyte is a compound, example glucose, urea, drug, pesticide, whose concentration has to be measured. Biosensors basically involve the quantitative analysis of various substances by converting their biological actions into measurable signals. A great majority of biosensors have immobilized enzymes. The performance of the biosensors is mostly dependent on the specificity and sensitivity of the biological reactions, besides the stability of the enzyme. Now let's know about the general features of biosensors. A biosensor has two distinct components. First is biological component, enzyme cells. Second is physical components like transducer, amplifier, etc. The biological component recognizes and interacts with the analyst to produce a physical change that can be detected by transducer. In practice, biological material is appropriately immobilized onto the transducer and the so prepared biosensors can be repeatedly used several times for the for a long period. Now let's know about principle of a biosensor. The desired biological material is immobilized by conventional methods, physical or membrane entrapment, non-covalent or covalent binding. This immobilized biological material is in intimate contact with the transducer. The analyte binds to the biological material to form a bound analyte, which in turn produces the electronic response that can be measured. In some instances, the analyte is converted to a product which may be associated with the release of heat, gas, that is oxygen, electrons, and hydrogen ions. The transducer can convert the product linked changes into electrical signals which can be amplified and measured. Now let's know about types of biosensors. There are several types of biosensors based on the sensor devices. and the type of biological materials used. Now let's know about the electrochemical biosensors. Electrochemical biosensors are simple devices based on the measurement of electric current, ionic or conductance changes carried out by bioelectrodes. Now let's know about metric biosensors. Amperometric biosensors. These biosensors are based on the movement of electrons, that is determination of electric current as a result of enzyme catalyzed redox reactions. Normally, a constant voltage passes between the electrodes, which can be determined in an enzymatic reaction that occurs. The substrate or product can be transferred an electron with the electrode surface to be oxidized or reduced. This results in an altered current flow that can be measured. The magnitude of the current is proportional to the substrate concentration. Clark oxygen electrode, which determines reduction of oxygen, is the simplest form of amperometric biosensor. Determination of glucose by glucose oxidase is a good example. In the first generation amperometric biosensors, there is a direct transfer of the electrons released to the electrode, which may pose some practical difficulties. A second generation amperometric biosensors have been isolated and developed, wherein a mediator takes off the electrons and then transfers them to electrode. These biosensors, however, are yet to become popular. Now let's know about blood glucose biosensor. It is a good example of atmospheric biosensors widely used throughout the world by diabetic patients. Blood glucose biosensor looks like a watch pen and has a single use disposable electrode consisting of a Ag or AgCl reference electrode and a carbon working electrode with glucose oxidase and a derivative of ferrocene as a mediator. The electrodes are covered with hydrophilic mesh gauze for even spreading of a blood drop. The disposable test strips sealed in aluminum foil have a shelf life of around 6 months. An amperometric biosensor for assessing the freshness of fish has been developed. The accumulation of inosine and hypoxanthine in relation to the other nucleotides indicates freshness of fish, how long dead and stored. A biosensor utilizing immobilized nucleosides, phosphorylase and xanthine oxidase over an electrode has been developed for this purpose. Now let's know about potentiometric biosensors. In these biosensors, changes in ionic concentrations are determined by use of ionic selective electrodes. pH electrode is the most commonly used ion sensitive electrode since many enzymatic reactions involve the release or adsorption of hydrogen ions. The other important electrodes are ammonia selective and carbon dioxide selective electrodes. The potential difference obtained between the potentiometric electrode and reference electrode can be measured. It is proportional to the concentration of the substrate. The major limitation of potentiometric biosensors is the sensitivity of enzymes to ionic concentrations such as H plus and NH4 plus. Now let's know about ion selective field effective transistors. ISFET are the low cost devices that can be measured for miniaturization of potentiometric biosensors. A good example of is an ISFET biosensor used to monitor intramyocardial pH during open heart surgery. Now let's know about conduct time conduct time trick biosensors. There are several reactions in the biological systems that bring about changes in the ionic species. These ionic species alter the electrical conductivity which can be measured. A good example of conductimetric biosensor is the area biosensor utilizing immobilized urease. Urease catalyzed the reaction. Let's know about the reaction. NH2 carbon 2 NH2 double bond oxygen plus 3 H2O in presence of urease gives rise to 2NH4 plus HCO3 minus plus OH minus. This reaction is associated with drastic alteration in ionic concentration which can be used for monitoring urea concentration. In fact, urea biosensors are very successfully used during dialysis and renal surgery. Now let's know about thermometric biosensors. Several biological reactions are associated with the production of heat, and this forms the basics of thermometric biosensors. They are more commonly referred to as thermal biosensors or calorimetric biosensors. It consists of a heat insulated box fitted with heat exchanger. The reaction takes place in a small enzyme packed bed reactor. As the substrate enters the bed, it gets converted to a product and heat is generated. The difference in the temperature between the substrate and the product is measured by thermistors. Even a small change in the temperature can be detected by thermal biosensors. Thermometric biosensors are in use for the estimation of serum cholesterol. When the cholesterol gets oxidized by the enzyme cholesterol oxidase, heat is generated which can be measured. Likewise, enzymes of glucose, that is enzyme glucose oxidase, urea, that is enzyme urease, uric acid, here enzyme is uricase, penicillin G, here enzyme is beta lactamase, can done by these biosensors. In general, their utility is however limited. Thermometric biosensors can be used as a part of enzyme-linked immunoassay, that is ELISA. In short, ELISA, ELISA. And the new technique is referred to as thermometric ELISA, TELISA. Now let's know about optical biosensor. Optical biosensor Optical biosensors are the devices that utilizes the principle of optimal measurements, absorbance, fluorescence, chemiluminescence, etc. They employ the use of the fiber optics and optoelectronic transducers. The word optrode is representing the condensation of the words optical and electrode is commonly used. Optical biosensors primarily involve enzymes and antibodies as the transducing elements. Optical biosensors allow a safe, non-electrical remote sensing of materials. Another advantage is that these biosensors usually do not require reference sensors as the comparative signal can be generated using the same source of light as the sampling sensor. Some of the important optical biosensors we will now discuss. First, fiber optical lactate biosensor. Its working is based on the measurement of changes in molecular oxygen concentration by determining the quencing effect of oxygen on a fluorescent dye. The amount of fluorescence generated by the dye film is dependent on the oxygen. This is because oxygen has a quenching effect on the fluorescence. As the concentration of lactate in the reaction mixture increases, oxygen is utilized and consequently there is a proportionate increase in the quenching effect. The result is that there is an increase in the fluorescent output which can be measured. Now let's know about optical biosensors for blood glucose. Estimation of blood glucose is very important for monitoring of diabetes. A simple technique involving paper strips impregnated with reagents is used for this purpose. The strips contain glucose oxidase, horseradish peroxidase, and a chromosome. Example toluidine. Glucose in presence of glucose oxidase gives rise to gluconic acid plus H2O2. Chromosome plus 2H2O2 in presence of peroxidase gives rise to color dye plus 2H2O. The intensity of the color of the dye can be measured by using a portable reflectance meter. Glucose strip production is a very big industry worldwide. Colorimetric test strips of cellulose coated with appropriate enzymes and reagents are in use for the estimation of several blood and urine parameters. Let's know about luminescent biosensors to detect urinary infections. The microorganisms in the urine causing urinary tract infections can be detected by employing luminescent biosensors. For this purpose, the immobilized enzyme, namely luciferase, is used. The microorganisms on lysis release ATP, which can be detected by the reaction. Let's know about the reaction. Luciferin plus ATP plus oxygen in presence of luciferase gives rise to oxyluciferin plus carbon dioxide plus AMP plus pyrophosphate plus light. Other optical biosensors. Let's know about the other optical biosensors. Optical fiber sensing devices are in use for measuring pH, PCO2 plus P oxygen in critical care and surgical monitoring. Now let's know about piezoelectric biosensor. Piezoelectric biosensors are based on the principle of acoustic, that is sound vibrations, since they are also called as acoustic biosensors. Piezoelectric crystals form the basis of these biosensors. The crystals with positive and negative charges vibrate with characteristic frequencies. Adsorption of certain molecules on the crystal surface alters the resonance frequencies, which can be measured by electronic devices. Enzymes with gaseous substrates or inhibitors can also be attached to these crystals. A piezoelectric biosensor for organophosphorus insecticide has been developed incorporating acetylcholine esterase. Likewise, a biosensor for formaldehyde has been developed by incorporating formaldehyde dehydrogenase. A biosensor for cocaine in gas phase has been created by attaching cocaine antibodies to the surface of piezoelectric crystals. Now let's know about the limitations of piezoelectric biosensors. It is very difficult to use these biosensors to determine substances in solution. This is because the crystals may cease to oscillate completely in the viscous liquids. Now let's know about the whole cell biosensors. Whole cell biosensors are particularly useful for multiple step or cofactor requiring reactions. These biosensors may employ live or dead microbial cells. Now let's know about the advantages of microbial cell biosensors. The microbial cells are cheaper with longer half-lives. Further, they are less sensitive to variations in pH and temperature compared to isolated enzymes. Now let's know about the limitations of microbial cell biosensors. The whole cells in general require longer period for catalysis. In addition, the specificity and sensitivity of whole cell biosensors may be lower compared to that of enzymes. Now let's know about the immunobiosensors. Immunobiosensors or immunochemical biosensors work on the principle of immunological specificity. coupled with measurement based on amperometric or potentiometric biosensors. There are several possible configurations for immunobiosensors and some of them are now we discussed. Now dis- we will discuss here. First is an immobilized antibody to which antigen can directly bind. Second is an immobilized antigen that binds to antibody which in turn can bind to a free second antigen. Third is an antibody bound to immobilize antigen which can partially released by competing with free antigen. Fourth is an immobilized antibody binding free antigen and enzyme labeled antigen in competition. For the biosensors, 1,3 piezoelectric devices can be used. The immunobiosensors use enzyme are the most commonly used. These biosensors employ thermometric or amperometric devices. The activity of the enzymes bound to immunobiosensors is dependent on the relative concentrations of the labeled and unlabeled antigens. The concentration of the unlabeled antigen can be determined by assaying the enzyme activity. Now let's know about application of biosensors. Biosensors have become very popular in recent years. They are widely used in various fields. Biosensors are small in size and can be easily handled. They are specific and sensitive and work in a cost effective manner. The tentative market share of biosensor application we now discussed. Some of the important applications of biosensors are now we will discuss. Applications in medicine and health. First is application in medicine and health. Biosensors are successfully used for the quantitative estimation of several biological important substances in body fluids. Example glucose, cholesterol, urea, etc. Glucose biosensor is a bio boon for diabetic patients for regular monitoring of blood glucose, blood gas monitoring for pH, PCO, and P oxygen is carried out during critical care and surgical monitoring of patients. Mutagenicity of several chemicals can be determined by using biosensors. Several toxic compounds produced in the body can also be detected. Now let's know about the applications in industry. Biosensors can be used for monitoring of fermentation products and estimation of various ions. Thus, biosensors help for improving the fermentation conditions for a better yield. Nowadays, biosensors are employed to measure the odor and freshness of foods. For instance, freshness of stored fish can be detected by ATPase. ATP is not found in spoiled fish and this can be detected by using ATPase. One pharmaceutical compound has developed immobilized cholesterol oxidase system for measurement of cholesterol concentration in foods. Example butter. Now let's know about the application in pollution control. Biosensors are very useful, very helpful to monitor environmental pollution. The concentration of pesticides and the biological oxygen demand can be measured by biosensors. Several environmental pollutants can be evaluated for their mutagenicity by employing biosensors. Now let's know about the applications in military. Biosensors have been developed to detect the toxic gases and other chemical agents during war, used during war. Now let's know about the immobilized enzymes and cells therapeutic applications. The industrial and analytical applications of the immobilized enzymes have been now we'll discuss the therapeutic applications are dealt with here. There are several limitations for direct use of enzymes for therapeutic purposes. These include the poor availability of the enzyme at the site of action, sensitivity to natural inhibitors, degradation by endogenous proteases, and immunogenicity of certain enzymes. First point, let's know about some examples. First is immobilized streptokinase and urokinase on cephalics can be used for the treatment of thrombosis. Second is some success has been reported in the treatment of inborn errors by employing immobilized enzymes, example phenylalanine hydroxylase to treat phenylketonuria. Lysosomal alpha 1,4 glucosidase to correct type 2 glycogen storage disease, POPS disease. Now let's know about the improved drug delivery by using immobilized enzymes. Urea urease modulated system. Let's know about urease modulated system. By using immobilized urease enzyme along with the substrate urea, the drug delivery can be enhanced as urease plights urea. There is an increase in the pH due to the formation of ammonium hydroxide. The drug located in a pH sensitive bioerodible polymer can be effectively released for its action. Now let's know about glucose oxidase glucose modulated system. For insulin delivery to the human body, a bioerodible polymer system containing insulin has been developed. The delivery of insulin can be modulated by immobilized glucose oxidase enzyme. As glucose oxidase acts on glucose, gluconic acid is produced which lowers the pH. The lower pH in turn causes the release of insulin from the bioerodible polymeric system. Now let's know about the immobilization of artificial cells. An artificial cell primary consists of a spherical semi-permeable membrane with comparable dimensions of a living cell. The biological materials such as enzymes enclosed within the artificial cells can be immobilized. The so immobilized compact artificial cells can function as artificial organs. The important artificial organs constructed include artificial kidney, artificial liver, blood detoxifiers, and immunosorbents. This functioning is, however, very limited. Multi-enzyme system can be immobilized in the form of artificial cells for the conversion of a substrate to a product through series of reactions. Finally finished. I have finished enzyme technology. Any questions regarding this enzyme technology, you can ask me in the comment box. And please kindly subscribe my channel to know more topics of biotechnology and get knowledge. Thank you.